Okay, so we all kind of remember that basic idea from biology class, right?
Right.
DNA makes RNA, RNA makes protein,
Right, the central dogma. Seems pretty straightforward.
like a neat little assembly line.
Yeah.
But what if that picture is way too simple? What if cells have these like really clever, totally unexpected ways to mess with the instructions after the copy's been made?
Exactly. And that's what we're diving into today. We're looking at this really pivotal 1998 paper from Nature.
Hmm.
And this wasn't just, a small finding. It seriously changed how people thought about genes working. Andrew Fire and his team, they stumbled onto something pretty amazing in this tiny little worm, C. elegans.
A real curveball, you see.
Oh, absolutely, a scientific curveball.
So our mission for this deep dive is to unpack that key discovery. This idea that double-stranded RNA, a specific type, has this incredibly powerful way of, well, interfering with genes.
Potent and specific.
And the crazy part is, nobody really predicted this, right? It wasn't part of the thinking at the time.
Not at all. It really came out of left field. This paper, technically a letter in Nature, which often means it's, really significant, urgent news.
Right.
It laid out how putting this double-stranded RNA into the worms could just shut down specific genes.
Oh.
And even then, back in '98, you could see this was hinting at something fundamental, some biological process that was mostly a black box.
So for you listening, this is a great look at how sometimes the biggest discoveries pop up where you least expect them. I mean, a tiny worm experiment changing biology.
That's often how it works.
Okay, so before this big discovery, what was the main idea about how you could interfere with a gene? You mentioned antisense.
Yeah, antisense RNA. The thinking was pretty logical, really.
Yeah.
Genes get copied into messenger RNA, the mRNA.
The messenger, right?
That mRNA carries the instructions to build a protein. So antisense RNA is just a single strand of RNA designed to be the mirror image, complementary to a specific mRNA.
Okay.
The idea was you put this antisense strand into the cell, it finds its matching mRNA, sticks to it like a genetic Velcro,
Yeah, okay.
and that double-stranded bit couldn't be read by the protein-making machinery, so boom, gene silenced.
A direct block. One antisense molecule stops one mRNA molecule.
Seems reasonable.
It does seem reasonable.
But Fire's team found something way stronger.
Way, way stronger. They were working with a gene called UNC22 in the worms. It makes a muscle protein.
Ah, the twitching worms you mentioned.
Exactly. It's not essential for the worm to live, but if you mess with UNC22, the worms twitch. It's easy to see.
Convenient.
Very. So they purified single strands of RNA, both the sense strand, which matches the mRNA sequence, and the antisense strand, the mirror image.
Right.
They injected these into the worms, targeting UNC22. And surprisingly, even when they used a lot of this single-stranded RNA,
Yeah.
almost nothing happened. Minimal twitching, if any.
Huh. So the antisense idea, the main theory, wasn't really panning out in their hands.
Not nearly as well as expected.
Okay, here's the kicker then. What happened when they tried putting both strands in together, a mix? That's where things got wild. The mixture, which would naturally zip up to form double-stranded RNA, was, get this, at least 2 orders of magnitude more effective.
2 orders, so 100 times.
100 times, minimum, more effective than either single strand by itself. Even tiny amounts, like maybe 60,000 molecules of each strand per worm.
Which sounds like a lot, but in a whole worm.
Exactly. Gets diluted fast, especially as the embryo develops, starting with only about 500 cells. But even that tiny amount caused obvious twitching in the offspring.
Wow. Okay, that's not just better, that's a fundamentally different phenomenon.
Yeah.
100 times more potent. That just shatters the old antisense model.
Completely. It suggested something much more powerful is going on.
And you mentioned this effect lasted into the next generation. That's also weird, isn't it? mRNA doesn't usually stick around that long.
Exactly. mRNA is usually pretty transient, so the fact that this effect persisted, passed down to the progeny, was really puzzling. It hinted that this wasn't just simple blocking. The researchers themselves pointed this out. They started thinking maybe the structure of the RNA, the fact that it was double-stranded, was the key.
Okay, so they suspected the double-strand structure was the magic ingredient. How did they prove that? How did they confirm it wasn't just, something weird about the mix?
Good question. They did a couple of really important checks. First, they used gel electrophoresis. That's a standard lab technique to separate molecules.
Right. Separates by size and charge.
Exactly. And they confirmed that their mix of sense and antisense strands was mostly forming double-stranded RNA. Then, crucially, they cut that double-stranded RNA band out of the gel, purified it. and injected that purified dsRNA. It worked just as potently. That pretty much nailed it. The double-stranded structure itself was the active component.
Okay, solid confirmation. Double-stranded RNA is the trigger.
Mm.
But did the strands have to be perfectly paired before injection? Did they have to, like, pre-anneal them?
That's another interesting wrinkle. Pre-annealing them, letting them pair up nicely in the test tube first, definitely worked well. But it wasn't absolutely necessary. They found that just mixing the two single strands in a low-salt buffer, which isn't ideal for forming dsRNA outside the worm, or even injecting one strand right after the other, boom, boom.
Quickly.
Yeah, quickly, that could also work pretty well.
Huh. So the worm's own cellular environment could help them pair up.
It seemed like it, but, and this is key, if they waited too long, say more than an hour, between injecting the first strand and the second strand,
Then what?
the effect dropped way off, dramatically.
Oh, okay. So the single strands probably got chewed up or hidden away somehow if they were left alone for too long inside the cell?
That's the likely explanation, yeah. Cells have enzymes that degrade RNA, so the single strands are vulnerable if they don't find their partner quickly.
Makes sense. Okay, specificity. That's always a big question with things like this. You're dumping foreign RNA in. Could it just be causing a general cellular freakout, like a panic response that shuts down random genes?
Absolutely critical point, and they tested that directly. They tried injecting UNC22 single-stranded RNA along with some unrelated double-stranded RNA.
Did that boost the weak single-strand effect?
Nope. Didn't help. They also tried sticking unrelated dsRNA bits onto the end of a single-stranded UNC22 piece.
Yeah.
Don't boost either. The powerful silencing only happened when the double-stranded RNA sequence actually matched the target gene's sequence. Homologous, as they say.
Like the puzzle pieces fitting together.
Exactly. It had to match the gene it was supposed to silence.
So not a general panic, it's targeted. Did they try targeting other genes besides UNC22 to really hammer this home?
They did. They picked 3 other genes where they knew exactly what happens when you knock them out. UNC54 for muscle contraction, FEM1 for sperm production, and HLH1 for body shape and movement. And in every case, injecting the matching dsRNA caused the offspring to show the exact same problems you see in worms with a mutation in that specific gene.
Wow, like a perfect mimic of the genetic mutant.
Precisely. And again, the single strands for those genes didn't do much at all.
Okay, that's compelling evidence for specificity. But I think you mentioned there was one little hiccup, one exception.
Yeah, there's one interesting case. When they targeted a specific part of the UNC54 gene, they called it UNC54C, the worms didn't just have muscle problems. They actually stopped developing early on, embryonic or larval arrest.
Which is not what happens if you just knock out UNC54 normally.
Correct. The normal UNC54 mutants develop, they just have muscle issues. So the researchers looked closer at that UNC54C sequence. Turns out that specific bit codes for a part of the myosin protein that's really similar across a whole family of related myosin genes.
Ah, so it might have accidentally silenced other important myosin genes too. Off-target effects.
That was their hypothesis, that the dsRNA for that conserved region was hitting not just UNC54, but also its close relatives, leading to a more severe outcome. So it highlighted a potential limitation. If your target sequence is too similar to other genes, you might get unintended silencing.
Right, a potential pitfall. But they said it wasn't common in their experiments overall.
Yeah, they emphasize it seems specific in most cases, but it's something to be aware of.
Okay, so we've got this powerful, mostly specific silencing by dsRNA. What about inside the worm, at the cellular level? Was every cell getting silenced equally?
Ah, another great question. They looked into that too. They used worms that were genetically engineered transgenic worms.
Yeah.
These worms were designed to make fluorescent proteins, GFP, the glowing green stuff from jellyfish.
Right, the reporter genes.
Exactly. Make GFP specifically in their muscle cells. So they injected dsRNA that targeted the GFP gene itself.
So they could literally see the silencing happen by looking for cells that stopped glowing.
Precisely. And they saw a big drop in the number of glowing cells. And in the cells that were affected, the glow was just gone. But interestingly, it wasn't an even pattern.
Not even. How so?
Well, it seemed like the muscle cells that are formed really early in the embryo were more easily silenced, and the effect stuck around as the worm grew. But muscle cells formed later during larval development. They seemed more resistant. And the vulval muscles, which formed quite late, seemed pretty much unaffected, even with high doses of the dsRNA.
Huh. So maybe the cell's age or how many times it had divided made a difference?
That's what they speculated. Something about the cell's developmental state, or maybe its cell cycle stage, seemed to influence how the RNA interference worked. It wasn't just a blanket effect on all cells.
Fascinating. More complexity. So the mechanism,
Yeah.
how is this actually working? Did the paper offer clues?
It did offer some crucial early clues, one big one. They tried using dsRNA that matched parts of the gene not found in the final messenger RNA, like introns.
The bits that get spliced out.
Exactly. Or the promoter region, the DNA sequence that turns the gene on.
Right.
Injecting dsRNA targeting those regions didn't work. No interference.
Okay, so that strongly suggests it's not messing with the DNA itself, or the act of copying DNA to RNA. It's happening after that, post-transcriptional.
That was the strong implication, yeah.
Yeah.
It seemed to be targeting the RNA molecule itself after it was made.
And did they actually see the target RNA disappearing?
Yes, this was a key finding. They looked at another gene, MEX3, which makes a lot of mRNA in the reproductive system and early embryos. When they injected MEX3 dsRNA, the amount of MEX3 mRNA in the worms plummeted. Sometimes it vanished completely.
Wow.
But, and this contrasts sharply with the old idea, when they injected just the MEX3 antisense RNA,
Yeah.
the mRNA levels didn't really drop.
Ah. So the dsRNA isn't just blocking the mRNA from being used, it's actually leading to its destruction.
That's exactly what the evidence pointed toward.
Yeah.
mRNA degradation, a much more decisive way to silence the gene.
Definitely. And then there was another really surprising thing. The effect could spread?
Oh, yeah, that was amazing. They found if they injected the dsRNA just into the worm's main body cavity, not even directly into the reproductive organs, it could still cause specific gene silencing in the offspring. And it affected the offspring from both arms of the gonad, suggesting the signal spread.
Wow.
Plus, injecting it into the body cavity of an adult could also silence genes in the adult's own body tissues, not just the germline.
So it's moving between cells. There must be some kind of transport system involved.
That's what it strongly suggested. Some way for the dsRNA, or maybe a signal generated by it, to travel across cell boundaries and have effects elsewhere in the worm. Really remarkable.
Okay. Just incredible findings all around. So let's pull back a bit. As the authors were wrapping up this 1998 paper, what did they see as the big takeaways, the main implications?
Well, first and foremost, they knew they'd hit on a fantastic new tool. Injecting dsRNA was suddenly this powerful, relatively easy way to figure out what genes did in C. elegans. You just knock down a gene you were curious about and see what happened to the worm.
Huge for researchers trying to understand unknown genes.
Absolutely. They did also mention the potential limitations we talked about, the off-target possibility with very similar genes.
Right, the Unki 54C example.
Exactly. And the idea that maybe some genes, perhaps ones expressed at really low levels or only in a few cells, might resist the interference.
Sure, makes sense. Did they think this was just a worm thing, or did they see broader potential?
Oh, they definitely speculated more broadly. They pointed out that weird gene silencing effects had already been seen in plants, things called co-suppression, which sometimes seemed linked to repetitive DNA sequences or plant viruses that make dsRNA. So they suggested this RNA interference, or RNAi as it came to be known, might be a widespread biological mechanism, not just worms, but maybe other invertebrates, maybe even vertebrates, plants.
So potentially a fundamental process across many forms of life.
That was the hint.
And the mechanism itself, beyond just destroying mRNA. Any early thoughts on how it was so potent?
They definitely ruled out the simple one-to-one blocking idea, the stoichiometry, because so few dsRNA molecules were needed to get such a big effect.
Right, the 100-fold difference.
Yeah. That smelled like some kind of amplification or catalytic process, where one dsRNA molecule could trigger the destruction of many mRNA targets.
Like an enzyme?
Possibly RNA-targeting enzymes, yeah. They also briefly considered if RNA could somehow directly affect the DNA, the chromatin structure, or transcription itself.
But the interim promoter results argued against that, didn't they?
Yeah, mostly yes. For the parts within the gene's coding sequence, it looked post-transcriptional. They noted if it did affect chromatin, it would be novel because it targeted sequences present in the final mRNA, not just regulatory DNA regions.
Okay.
And importantly, they proposed this whole system probably exists for a reason, a natural biological function.
Not just a lab trick.
Right. Maybe for regulating the worm's own genes normally. Physiological gene silencing. And the spreading effect. That pointed to an active RNA transport system in the worm. They were already thinking about the bigger biological picture.
Okay, let's try and distill this down. For you listening, the really big game-changing takeaways from this single paper were: 1, this massive surprise double-stranded RNA is insanely better at gene silencing than single-stranded.
Huge surprise.
2, it's remarkably specific. It hits the gene it's supposed to hit. 3, it seems to work by actively destroying the target messenger RNA after it's made.
Post-transcriptional degradation.
And 4, this effect isn't just stuck in one cell. It can actually move around the organism and even be passed to the next generation. That's just a whole new layer of gene control nobody saw coming.
It really is. It just blew the doors open on gene regulation research. It showed there was this whole other level of complexity of this RNA-based control system, and it highlights how crucial basic research, even in a tiny worm, can be. You never know what fundamental principles you might uncover.
Totally. So the lingering question, the thing to chew on after this deep dive.
Yeah.
What is it?
Well, maybe it's something like this. If a process we thought we understood reasonably well, like gene expression, had this incredibly powerful and elegant silencing mechanism hidden inside it, just waiting to be found,
Yeah.
what other fundamental biological processes, things we think are straightforward, might also have these kinds of surprising, maybe RNA-based control systems operating that we just haven't discovered yet?
That's a great question.
Or, flipping it around, now that we know about this natural gene silencing tool that cells use, how can understanding it better maybe lead to new ways to treat diseases or develop new biotechnologies? The paper itself hinted at the possibilities.
Really makes you think. And if you were intrigued and want to know more, definitely search for RNA interference or RNAi and look up the researchers. Veeren and Mello actually won the Nobel Prize for this work later on.
They did indeed. Well deserved.
This 1998 paper really did kickstart a revolution. We've only just scratched the surface here today. Thanks so much for joining us on this deep dive.